Hello friends, welcome back to another video tutorial from Shomu's Biology. And in this video lecture, we want to talk about the eukaryotic replication or the replication in eukaryotes. So eukaryotic replication is a completely different and complex process of replication compared to the prokaryotic DNA replication. So eukaryotic replication is the only thing where the prokaryotic and eukaryotic system differs a lot. While in case of transcription of eukaryotes and translation of eukaryotes with prokaryote comparison, they are not that different. But in DNA replication, it is totally different. The reason eukaryotic DNA replication is more complex is the eukaryotic DNA is more complex. It is made with so much material, so many information, right? It is composed of important genes and the genes which are not coding for any proteins, but it is all present in the DNA. The genome is huge. So for the eukaryotic replication, this is the first lecture regarding the eukaryotic replication initiation. So initiation part of the eukaryotic replication is probably the most important part that you need to understand very, very well. Okay. So first things first, let me give you a very quick basic idea about the DNA replication. You all know that the DNA replication means simply making a copy of the DNA. And the process of DNA replication is semi-conservative. That is in prokaryotes as well as in eukaryotes. That means in case of DNA replication, one strand used as a template, the old strand, parental strand used as a template, and a new daughter strand is produced utilizing that parent strand as a template. And both the strands are utilized as a template to make two new daughter strands. Thus, two DNA molecules can be formed from one DNA. So this is a very basic idea which is true for both prokaryotic DNA replication as well as in eukaryotic DNA replication. But in eukaryotic DNA replication, there are a few more important things, more involvement of proteins that we are going to see. And what are the proteins that are involved in eukaryotic DNA replication? The major protein is the DNA polymerase, which actually is involved in the job of polymerization of the DNA. So DNA polymerase is the very first one. There are different types of DNA polymerase enzymes. We are going to talk about that alpha, beta, gamma, delta, epsilon, and they have their different functions. We are going to talk about that in a moment. And apart from the DNA polymerase, there are other proteins which are also involved in the process of eukaryotic DNA replication, and those are RPA. Okay, you can easily search the full forms online, so I'm not going to talk about that. But the RPA's job is like a single-stranded binding protein. There is a protein named RFC which acts as a clamp loader. If you recall the prokaryotic DNA replication, we know there is a clamp loader protein which helps to load the beta clamp onto the template DNA so that the DNA synthesis can be continuous. So in this case of eukaryotes, the name is RFC. Then there are ORC, which is known as origin recognition complex. Okay, so this is another protein complex. Generally, these proteins, they are not single structure or single subunit proteins. There are, most of them are multi-subunit, at least two subunit proteins. So similarly, origin recognition complex is a protein with multiple subunits, and they recognize the origin of DNA replication in eukaryotes. And there are this MCM protein. The full form is mini chromosome maintenance complex protein. Okay, so these proteins play very important role, very crucial role in the process of DNA replication. Apart from all these proteins, there are also other proteins like CTD proteins. We're going to talk about that later. And there are also proteins like CDC proteins, which we saw in case of cell cycle regulation. We're going to talk about that later as well. And another group of important protein is this CDK or cyclin dependent kinase, and DDK, which is known as DDF4 dependent kinase proteins. So these kinase proteins are also very, very important in order to allow the eukaryotic DNA replication to shift from the G1 phase into the S phase. So the CDK and DDK does the job. They do the job of ultimately telling the cell that it's the time to move from G1 phase to the S phase. Okay, so there are so many things and so many complicated topics about the eukaryotic replication, but the very basic idea of DNA replication remains the same, and that is the properties. If I talk about the DNA replication properties in here, so the properties means in this case also the DNA replication take place from 5' end to the 3' end of the newly replicating DNA. And in eukaryotes, 3000 base pairs are added. 3000 bases are added per minute. This is another statistics. And the third thing that is unique about the eukaryotic DNA replication is that because the eukaryotic DNA is not free like the prokaryotic DNA, because the prokaryotic DNA is kind of naked. They're not wrapped around any histone or any other proteins, but eukaryotic DNA is wrapped around histone. So in order to get an access to that DNA, we need to unwrap the DNA from the histone. So this is very, very important. During the process of DNA replication, even before the DNA replication, the DNA must be unwrapped from the histone completely. And the second thing is that as we are making a new version, a new copy of the DNA, we are going to require more nucleosome proteins, more histone proteins. So the histone proteins are also produced in the cytosol, and there are some escorting protein which brings those histone protein from the cytosol inside the nucleus through the nuclear pore complex. And there are also other assembly proteins which are recruited inside the nucleus to load the newly synthesized DNA onto the histone octamer. So these steps are additional in case of eukaryotic DNA replication, which is not present in prokaryotic DNA replication. So here I am going to talk about the initiation process or the start process of the DNA replication in eukaryotes. And in the start process, we are going to see there are different proteins involved in the process. And even at the beginning, what is the idea? You know that in G1 phase, the cell is making all the proteins which are important for the DNA replication purpose. So G1 stage is for the preparation phase. Okay, so we can call it as a preparatory phase. Okay, here the preparation takes place. And then S phase is the phase where actually DNA replication takes place inside the cell. But the signal to go from G1 phase to S phase is provided by the CDK and DDK together, because the presence of CDK and the concentration of CDK, whether the CDK is high or low, that actually dictates whether the process will continue to the next phase or not. Because in the G1 phase, they are going to make all the proteins necessary. They are going to make DNA polymerases. They are going to produce the RP, RFC, ORC, NCM, all these proteins, CDT, all these proteins are prepared. But in the when the protein is prepared, then the cell needs to give a signal so that the cell move from the G1 phase to this phase, and that is done by this CDK and DDK protein complex. How exactly? Let me give you a simple idea. If you think about the eukaryotic DNA replication, we can divide them in different stages. So we can go simply like the stage 1 is the pre-initiation complex. Okay, and second one is the initiation. These are the two steps of an initiation process. So if you are talking about only initiation of eukaryotic DNA replication or DNA replication in eukaryotes, then there are two steps: pre-initiation and initiation. Pre-initiation is the time where most of the proteins are synthesized. So let me write it here. Most of the protein synthesis are done, and also including the DNA polymerase synthesis is done. In the initiation phase, those proteins are utilized and the signal is received from CDK that provides the idea to the cell that now it's time to move from G1 phase to the S phase. And how exactly it's done? Because the thing is that CDK concentration regulates it. How it regulates it? Let's assume the CDK concentration is low. If the CDK concentration is low, CDK can do two of this important job. One is that, let me write them here. One is the formation of pre-initiation complex. Okay, and second thing is activation of existing initiation complex. Okay, so these are the two things. Both can be activated by the CDK. Okay, so CDK can initiate the formation of pre-initiation complex in eukaryotic DNA replication. CDK can also activate the existing pre-initiation complex and convert it into the initiation complex, and thus the cell can move from G1 to this phase. But when the CDK concentration is low, then what CDK does is that CDK only signals the formation of new pre-initiation complex, but it prevents the activation of that pre-initiation complex into the initiation complex. When the CDK activity increases inside the cell, when the CDK activity is high inside the cell, then the CDK signals the conversion of the already existing pre-initiation complex into initiation complex, while it will inhibit any further formation of new pre-initiation complex. Okay, so I think you got the idea. So what we need is that at the G1 phase, if you think about what happens in the G1 phase, the CDK concentration was low. So what due to the CDK concentration low, the formation of new pre-initiation complex begins in the G1 phase. Then as the cell is processing, as the cell is making all the proteins necessary for DNA replication, the cell gets the signal, means the CDK concentration is increasing, because I told you in G1 phase, this is the preparation phase, CDKs are prepared and the concentration start to rise from G1 towards S phase. And when the CDK concentration increases inside the cell, then the high CDK concentration activate the already existing pre-initiation complex into the initiation complex, and then this process takes place in the S phase. So now we call it as the S phase, because now the initiation complex is formed, and when the initiation complex is formed, then only the DNA replication in eukaryote can properly begin. We are going to talk about the steps of this initiation complex formation, step of the process of DNA replication in eukaryotes in a moment. But before going into there, this is the regulation that you need to have a very clear idea about even before going into the detail of replication itself. So now, as you know how it is regulated by CDK and DDK, the second thing that we want to talk about is the major enzyme, the key enzyme for DNA replication. And the key enzyme is DNA polymerase. And there are different types of DNA polymerase. Like in case of prokaryotes, there are different types of DNA polymerase. Polymerase 1, 2, 3, like that. In eukaryotes, there are polymerase alpha, beta, gamma, delta, epsilon. And now there are different debates regarding what polymerase is doing, what function, because the research is going on and the new data is coming almost every single year. So what I'm going to tell you is kind of established fact till now, is that all this alpha, beta, gamma, delta, epsilon, they don't have the same job. What alpha polymerase do is that alpha polymerase has two different kind of subunits. It has two jobs. One, it has the job of primase. So it does the job of primase, means addition of primer, and it also adds the first nucleotide, first deoxyribonucleotide as well. For the initial 20 nucleotide addition is also done by the alpha polymerase in both the strands, leading and lagging strands. While the beta polymerase, if you think about the beta polymerase, is only involved in the process of DNA repair. Okay, this is only for DNA repair mechanisms. Then there is this gamma. The gamma polymerase of eukaryote, the eukaryotic gamma polymerase, involved in the DNA replication in mitochondrial DNA. So mitochondrial DNA replication is conducted by the DNA polymerase gamma. And now the last two polymerases we have the delta and we have the epsilon. These are the two polymerases which are involved in the process of actual polymerization of the DNA. By actual polymerization, I mean the delta one is involved in the lagging strand synthesis. Epsilon is involved in the leading strand synthesis. Now in many places you may find that the delta is also written in case of the leading strand synthesis, because there are research going on and there are both this theory out there that this delta can synthesize both lagging as well as leading strand. It has the capability. But in normal situation where everything is okay for the cell always go with the epsilon polymerase for the leading strand synthesis. Although delta has the capability of synthesizing both lagging as well as leading strand. Okay, so I make this thing clear in here, because you may find this thing. This is not wrong. The idea is kept here. Okay, so this is the idea, delta and epsilon. But for all this, throughout this series of DNA replication topics, we are going to assume that the delta is used for the lagging strand synthesis, epsilon polymerase is used for the leading strand synthesis. Okay, that's the idea. And because the treatment of DNA synthesis in leading strand and lagging strands are different, just like in case of prokaryotes. Similarly, the leading strand synthesis is done continuously without any sort of gap or any fragments, while in the lagging strand synthesis, there are small fragments are produced known as Okazaki fragments. And here comes another interesting idea, that is the Okazaki fragment length for the prokaryotes are much bigger, but the Okazaki fragment length for the eukaryotic DNA replication is much smaller. That's another important difference between prokaryotic DNA replication and eukaryotic DNA replication. Okay, hopefully you get this idea clear now. So these are the basics, these are the basics of the process of DNA replication in eukaryotes. And once you know about all the name of these proteins, there are a few more protein name is going to come in the future times. So I have my whole board filled up, so I'll clean it up. And we are going to talk about the starting of the DNA replication in eukaryotes. That means the formation of pre-initiation complex and then the formation of initiation complex and how it is done. That's what we're going to talk about now. Okay, so my board is clear, and also I've written the name of some more proteins. Okay, it's very difficult to put all the name of the proteins in the mind while explaining, so I put it very clearly in here, and I'm going to talk about individually each of them. So our idea here is to understand the process of initiation of DNA replication in eukaryotes. So what happens exactly during eukaryotic replication initiation? See, the first things first, what I should draw here is the DNA itself. So there is a origin of replication always. In case of prokaryotic DNA, there is a origin of replication, and eukaryotes there is also origin of replication. Let's say this is the origin of replication, okay? And this origin of replication site must be recognized by a protein complex. In this case, that is ORC, origin recognition complex. The very first protein that involved in here is the ORC, origin recognition complex. And once origin recognition complex bind to the origin recognition region of the DNA, then after that the sequential events start to begin. This process, they're making the DNA ready. Means at this point we are talking about the G1 phase, where all the proteins are produced in the G1 phase. Okay, every single protein written in here, they are already produced in the G1 phase. And the pre-initiation complex is about to form. So we are talking about G1 phase now. Okay, so in G1 phase, origin recognition complex recognizes the origin. Now they recruit other proteins, other two proteins. What are those proteins? CDC6 and CTD1. Okay, these proteins are known as helicase loader proteins. Okay, because normally in this case we need to have helicase loader protein that will load the helicase here. And the name of the helicase in case of eukaryotic DNA replication here is MCM protein, MCM2 to MCM7 complex proteins. They are known as the helicase, and they have the similar job of helicase activity. That means they will load itself between two strands of the DNA, start to move through the DNA and separating the strands from each other. So ORC recruits CDC6. Remember, ORC recruits here CDC6 and CDT1. Okay, and they recruit MCM proteins. MCM means helicase. Remember, now when helicase is loaded, then what will happen here is that this origin will start to melt. Means the helicase is going to unwind the DNA. They are going to open the strands from each other. This is the time where we have this kind of structure. We have a bubble forming at this point, okay, like this. Because think about eukaryotic DNA replication. In prokaryotic DNA replication, there is always one origin of replication, okay, and from that one origin, one replication bubble is formed, and two replication form move towards opposite direction, one clockwise, another one is anti-clockwise, and they meet in the termination site. While in eukaryotes, there are multiple origin of replication. So this kind of replication bubble or melting of the DNA will be visible in many different places of that the complete length of the genome. So here what we can see that once CDC6, CTD1, they recruits MCM, which is helicase, they start unwinding the DNA. And now the template DNA strand, the bases are open and single-stranded binding proteins will bind here. The name of the protein we already talked about earlier. So those proteins are already, they are blocking everything. But this is again in the G1 phase. Remember, till this point we are talking about the G1 phase. Now once the helicase is loaded, now it's time to recruit polymerase. Which polymerase? Polymerase alpha. The eukaryotic DNA polymerase alpha. And the eukaryotic DNA polymerase alpha is versatile because it can act as the primase enzyme and can add the primer. And not only it can only add primer, but also it initiates and elongates that primer by adding deoxynucleotide sequences, at least till 20 nucleotide sequence. So at this moment when the helicase is loaded, this is the time where the CDK checks where everything is okay or not. And when the CDK concentration increases, remember the CDK concentration increases in the cell, then this G1 stage of the cell cycle shifted to the S phase of the cell cycle. And when it is shifted to the S phase, at this moment, there are complex of protein that are activated, that are added to this replication machinery. So this was pre-initiation complex, but once the CDK activity increases inside the cell, CDK concentration increases the cell, they will recruit CDC45, MCM and GINS genes. CDC45, MCM and genes, these protein complex together, once loaded, that means the S phase begins. Okay, and at this point, once all these proteins are loaded, because they are marking the site where the polymerase alpha will come and bind, and polymerase alpha will add the primer. This is very, very important. So I'm not going to draw these complex proteins. Let's assume that these are the complex of proteins that are present there, and now they will recruit the polymerase alpha. And what polymerase alpha will do is that polymerase alpha firstly will add a primer sequence. Okay, and remember, if I am drawing this primer here, so obviously it is 5 prime and we are going 3 prime. So this one is 3 prime, this is 5 prime of the mother strands. So what we can clearly see is that primer is added, and not only the primer, remember I told you it's very unique because this polymerase alpha for eukaryotic DNA polymerase, they also add nucleotide sequences up to 20 nucleotide here, the alpha. Okay, that process is done. And once that process is done in the leading strand, so let's assume this is going to be the leading strand, then this is going to be the lagging strand. So again, the primer is added for the lagging strand in this side. Okay, an extension is also done here as well by the polymerase alpha. And now once this thing is done, then the polymerase delta and epsilon will be loaded. Polymerase delta and epsilon will be loaded, and polymerase delta epsilon will be loaded where exactly? Depending upon which strand is going to be leading and which strand is going to be lagging. Now here is another concept check. In case of prokaryotic DNA replication, there is only one enzyme, that is DNA polymerase 3, that replicates both the strands together simultaneously. But in case of eukaryotic DNA replication, there are 2 separate dedicated DNA polymerase enzymes to replicate the 2 different strands. Okay, so in this case, we are not going to talk about how they are going to move, because there are 2 separate polymerases. And as I told you, there are although confusions, but DNA polymerase delta most of the time here is synthesizing the lagging strand, and DNA polymerase epsilon of eukaryotic polymerase is involved in the synthesis of leading strand. So this is where I can draw the epsilon polymerase loaded. This is where I can draw the delta polymerase is loaded. This is leading strand, this is lagging strand. And again, like in the lagging strand, what will happen, there will be multiple sites where the primer must be added by the DNA polymerase alpha, and then the process may continue. So this is how they will begin the process. And once the polymerase epsilon and delta are loaded onto their destination strands, then there is another protein known as RFC. What we mean by this protein RFC? This RFC is the replication factor, or we can say replication factor C. This replication factor C, what is the job? Replication factor C recognizes the interaction and the region between the primer and the nucleotide. The primer and the nucleotide. This is very important. You know why? Because this is red colored is the primer, and this is the nucleotide, which is deoxyribonucleotide, which is also added by the DNA polymerase alpha. Sorry. So at this moment, what happens is that this RFC, or this replication factor C, always find out the junctions between the primer and the template DNA, and the primer with the nucleotide sequences, which is extended by the polymerase alpha. And this is where they are going to load a protein known as pCNA, or we can also write it as a pCNA ring. Okay, why we call it pCNA ring? Just like the clamp, the sliding beta clamp. In case of prokaryotic DNA replication, we saw beta clamp protein is there. Clamp loader loads the beta clamp protein in the template DNA strands, so that the polymerase can hold on to the template DNA and keep on adding nucleotide sequences, and that increases the processivity of the DNA replication capability of the DNA polymerases. The same thing happens here. In this case, the ring is made with this pCNA ring, and that is loaded by this RFC or replication factor C. That helps to add the pCNA ring in both the strands, and once the pCNA ring is needed for both, by the epsilon polymerase as well as by the delta polymerase. Because without this pCNA ring, without this kind of hook like structures, the polymerase may not be able to add a long stretch of DNA sequence. It will fail and it will fall off from the DNA. So the pCNA is loaded, and that is recognized by the RFC. They help in loading the pCNA. Once pCNA ring is loaded, now it's time for the whole machinery, whole DNA replication machinery to move towards their direction. In the leading strand, the movement will begin from 5' end towards the 3' end of the newly synthesizing strand. In the lagging strand also, the 5' to the 3' end of the newly synthesizing strand. But in the leading strand, we'll have a continuous stretch of lengthy DNA synthesis with only one primer here. But in case of eukaryotes, if you think about it, in eukaryotes there are so many different replication bubbles in different places. So for in each of these replication bubble, even in the leading strands, we are going to find out like for each replication bubble in the leading strand, at least one primer will be present. Okay, so in case of eukaryotic DNA, at the end of the replication, not only the lagging strand where we have the multiple primers, but the leading strand also contains multiple primers due to the multiple replication bubble formation. So at the end of the replication, what we need to do, we need to cut all those primers out and we need to fill the gap and we need to seal the nick. So again, in this case of sealing, the nick is done by the DNA ligase, but cutting the primer out can be done by two different methods, which we'll talk in the process of termination later on. But right now, get this idea: once the polymerase epsilon and polymerase delta are loaded, then they will continue. PCNA once loaded, then only they will continue to move because helicase is there. Okay, so helicase start to unwind the DNA from each other, separate the strands from each other, and so as the epsilon and delta start adding nucleotides, deoxyribonucleotides, and they continue to move toward their destination, that is 5' to 3' of the newly synthesizing strands. This is the initiation. Okay, and this process of the origin escape, this we can call it as a DNA replication origin escape in eukaryotic DNA replication, and that is done in the S phase, while the pre-initiation complex formation is done in the G1 phase. Keep this thing in your mind. And in the G1 phase, even before all this process is done, one extra step was performed, which I told earlier, is the unwrapping of the DNA from the histone octamer, so that the DNA is totally free to be utilized for the DNA replication. That is another unique point about the eukaryotic DNA replication. So there are plenty name of the different proteins, and there are plenty of proteins involved in the initiation process of the DNA replication in eukaryotes. So that is kind of overall summary of eukaryotic replication initiation. If you like this video, please hit the like button, share this video with your friends, subscribe to this channel to get the next part of this video, that is the elongation of eukaryotic DNA replication as well as the termination of eukaryotic DNA replication. Okay, so if you want to learn about the DNA replication in eukaryotes in details, watch this DNA replication in eukaryotes series in my channel. Thank you. We'll see you in the next video. Bye.